Now let's look at the last section, which is the nucleic acids. Nucleic acids are it's the macromolecule that is present for hereditary information. That is one role for this macromolecule. And there are two different kinds of nucleic acids, and one of them definitely has a hereditary role. The nucleic acids are long chains of units, and these units are called as nucleotides. So the monomer for the nucleic acid are nucleotides. Nucleotide is a complex molecule that has three components, and you can see in this image the three components. Each nucleotide has a central unit which is called as the sugar. Then it always has a unit that is called as the phosphate group. And lastly, it has a unit that is called as the nitrogenous base. Whether it's the nucleic acid called as deoxyribose or whether it's the ribose nucleic acid, they will always have the nitrogenous base and the phosphate. And what is different over here, I don't know if you can see it that closely, notice that the pentose sugar is slightly different in DNA and RNA. There is an oxygen missing in the sugar on carbon 2, and that gives the name deoxyribose. RNA, on the other hand, is a term for ribose nucleic acid. Another type of nucleic acid that you also will hear about is ATP, which is adenosine triphosphate. Adenosine triphosphate is the currency of the cell. So this one is not going to be needed for hereditary information. DNA is definitely for hereditary information. RNA can be hereditary and enzymatic. So it has two roles. It can work as an enzyme, and it can work as a hereditary information. Adenosine triphosphate is your currency of the cell. So these are the three important nucleotides that you need to know for this class. So let's go on to the next one. What is the structure of DNA? DNA is a double helical structure. So the double helical means that it has two strands. Double, you can see there are two ribbons, so there are two strands, and each of these strands are helically intertwined to give you a helical appearance. The sugar in the deoxyribose is definitely deoxy. The nucleotides are joined via a linkage, and these are called as the phosphodiester linkages. Links or bonds. So these are the phosphodiester bond, and these are formed because the sugar and the phosphate is linked up on each side. So the sugar phosphate gives this appearance of this template that I'm highlighting over here, and that is causing the links. Now in between the two ribbons, you see a region which is almost like the step of the ladder, and these steps are made by hydrogen bonds. These steps are they are not made by hydrogen, they are put together by hydrogen bond. They are made by the nitrogenous bases, and the nitrogenous bases will connect to each other through hydrogen bonds. The nitrogenous bases that are present in DNA are of two kinds. One kind is called as purine. Purine stands for double ring structure. Now look at the width of these two over here. Adenine and guanine both have two rings in them. So these two would be present in DNA. Besides that, it has one ring structure which is called as cytosine, and another one ring structure called as thiamine. The two one ring nucleotides are called as pyrimidines, and the two rings are called as purines. In order for the latter to have a uniform width, it was understood that one ring structure is pairing up with a two ring structure to give you this format. The one ring structure is adenine always binds with thymine and guanine always binds with cytosine. This is an important thing, and we will also learn this later under DNA, but I just wanted to introduce this purines and pyrimidines concept over here. So purines are your double ring, pyrimidines are your single ring. In the two that I've given you, your thiamine and cytosine are your pyrimidines, which are these ones over here. So that's a lot of color on this particular chart. Now, what is RNA? RNA, on the other hand, is ribose nucleic acid. RNA is always single-stranded. It is never found in double-strand. Sometimes you see folded forms, that is like an end that has folded on top of each other, but it is usually a single strand. Again, notice that the linkages are made by the phosphate sugar, phosphate sugar, phosphate sugar. So that also has a phosphate sugar backbone. The sugar is a ribose sugar as compared to the deoxyribose in DNA, and the only difference in the bases is that it does not has thiamine, and in place of thiamine it has uracil. So here is the uracil. So uracil is present in replacing thiamine. The other three bases would be the same. You would still have adenine and guanine as the purines, and you will have cytosine as the pyrimidine base, and then you would have the uracil as the RNA base. What is ATP? ATP stands for the term adenosine triphosphate. Adenosine triphosphate has 3 phosphate bonds, and these phosphate bonds are very high energy bond, and these high energy bonds are highly unstable, and when these bonds are split or broken during reaction, that releases tremendous amount of energy that is used by the cells for all kinds of function, whether it's your movement or whether you're chewing your food. So this concludes our nucleic acid exploration. We discussed DNA, RNA, and ATP.